Hello everybody, I'm Stephanie Longet. I'm a postdoctoral researcher at the University of Oxford, UK, and today I'm going to speak about the impact of short or long dosing intervals on antibody responses to Pfizer-BioNTech COVID-19 vaccine. SARS-CoV-2 emerged in December 2019 in China. It caused a pandemic. COVID-19 mitigation measures were implemented to reduce the spread of the virus and the impact of infections on populations. But of course, the quick development of COVID-19 vaccine was crucial in the control of the pandemic, at least in the countries where the vaccines are available. These vaccines were developed using various platforms. So now we have some mRNA vaccines, viral vector-based vaccine, and some inactivated vaccines. All around the world, some research groups analyze the protective immunity to COVID-19, including the PITCH Consortium, which is a consortium in the UK. The goal of the PITCH Consortium was to analyze antibody and cellular immune responses to COVID-19 following infection or vaccination in UK healthcare workers. The PITCH Consortium performed a prospective longitudinal cohort study in 5 sites: Oxford, Liverpool, Sheffield, Birmingham, and Newcastle. And there are more than 2,000 healthcare workers who were recruited to date. The PITCH Consortium wanted to answer 3 key questions. The first one, does previous infection impact on response to vaccination? For the following ones, they are related to UK government decision. Maybe some of you remember this, but in December 2020, the UK government decided to delay giving second doses in order to give more first doses to more people. But at that time, we didn't know anything about the impact of this delay on vaccine-induced immune responses. So the following questions were, what immunity do people have during an extended interval between doses? And does the immune response differ between long and short intervals? As you maybe know, T cells and antibodies work in combination to control viral infection. So there is the innate immunity and the adaptive immunity, which includes T cell responses and antibody responses. CD4 T cells orchestrate everything. CD4 T cell responses impact on CD8 T cell responses, which can lead to the clearance of infected cells. But CD4 T cell responses also impact on antibody responses. And today I'm going to focus on antibody responses and more particularly neutralizing antibodies. So they are the antibodies which can neutralize the virus before the virus can enter the cells. To analyze the antibody responses, first we used a multiplex immunoassay, so multiplex ELISA developed by Mesoscale Discovery. And the goal was to quantify the level of antigen-specific IgG antibodies. But in this assay, there were 9 antigens in the wells. There were SARS-CoV-2 spike, receptor binding domain and N, but also SARS-CoV-1 and MERS spike antigens, and the human seasonal coronavirus spike proteins. And the principle is like an indirect ELISA. So you have the antigens in the well, you add your plasma or your serum which contains some antibodies, and you detect this interaction using a labeled secondary antibody. And at the end, you can quantify the electric signal. We also analyze the neutralizing antibody responses. For this, we use an ACE2 inhibition assay developed by Mesoscale Discovery as well. So it's a surrogate neutralization assay. So the principle is quite similar to this ELISA. So basically, in the wells here, you have the variant spike proteins. And you add a label ACE2. So I remind you, ACE2 is a receptor which binds the virus, which binds SARS-CoV-2 through the spike protein. So here you add the label ACE2. You also add your samples, your plasma or your serum, which potentially contains some antibodies. And it's a competition assay. So the goal was to quantify the activity of antibodies able of blocking the binding of phase 2 to variant spike proteins. And finally, we also use a live virus neutralization assay. So here you have some cells. You infect the cells with SARS-CoV-2, or you can also infect the cells with SARS-CoV-2 mixed with the plasma or the serum, which can contain some antibodies. And if there are some antibodies, the rate of infection is reduced, and it's possible to quantify the reduction in the number of foci. The first goal of this study was to evaluate the impact of a SARS-CoV-2 pre-infection on antibody responses induced by one dose of Pfizer BioNTech COVID-19 vaccine. So for this, we analyzed the antibody responses 28 days following the first dose in SARS-CoV-2 pre-infected individuals versus naive individuals. Now I'm going to show you some selected results. Here you can see SARS-CoV-2 spike-specific IgG level. In black you have the naive participants. In red you have the SARS-CoV-2 pre-infected participants. Open circles show IgG level before vaccination, and shaded circles show IgG level following a first vaccine dose. And you also have another group in blue. They are naive participants who received 2 doses. And here you can see, following a third vaccine dose, there are higher SARS-CoV-2 spike-specific IgG level in plasma of SARS-CoV-2 pre-infected individuals compared to IgG level found in the plasma of naive individuals, but also compared to IgG levels in naive participants who received 2 doses of vaccine. We also observed that 1 dose of Pfizer BioNTech COVID-19 vaccine induced cross-reactive IgG responses to SARS-CoV-1 and MERS spike antigens, but also to human seasonal beta coronaviruses HKU1 and OC43. And these cross-reactive IgG responses were particularly observed in SARS-CoV-2 pre-infected individuals. The second goal of the study was to analyze the impact of dosing interval. So for this, the individuals received a second dose either between 2 and 5 weeks following the first dose, or between 6 and 14 weeks following the first dose. And we analyzed the antibody responses at various time points following the second dose, and also during the long interval. And again, we compare antibody responses in SARS-CoV-2 pre-infected individuals versus naive individuals. Now I'm going to show you some selected results. Here you can see SARS-CoV-2 spike-specific IgG levels in individuals who received the doses with a long interval. In gray you have the naive participants. In red you have the SARS-CoV-2 pre-infected participants. And here you have the various time points. And here you can see there is a decline of SARS-CoV-2 IgG levels during the long interval, and particularly in naive participants. But the second dose boosts SARS-CoV-2 IgG levels in both groups. We also analyzed the neutralizing antibody titers against the classical strain Victoria and some variants of concern like beta, gamma, and delta. We did that in naive participants who received the doses with a long interval. And here you can see there is also a decline of neutralizing antibody titers during the long interval, but the second dose boosts the neutralizing antibody titers against the classical strain and the variants of concern. And now the most important finding. We compare the impact of short versus long dosing interval on IgG levels following the second dose. Here you can see SARS-CoV-2 spike-specific IgG levels 28 days following the second dose. In gray you have the naive participants. In red you have the SARS-CoV-2 pre-infected participants. And you can see there are significantly higher spike-specific IgG levels in the plasma of individuals who receive the doses with a long interval compared to IgG level found in individuals who receive the doses with a short interval. And this significant difference is only observed in naive participants, and it's not observed in SARS-CoV-2 pre-infected participants. Similar results were observed for the neutralizing antibody titers against the Victoria strain and some variants of concern like alpha, beta, and gamma. And again, you can see there are higher neutralizing antibody titers in individuals who receive the doses with a long interval compared to the individual who receive the doses with a short interval. And this is only observed in naive participants and not in previously infected participants. As you can imagine, there is a follow-up of this study. So we also analyzed the antibody responses 3 months and 6 months following the second dose. And now we are starting analyzing the antibody responses following the third dose. Here you can see in gray the naive participants, in red the pre-infected participants, and it's a comparison long versus short dosing interval. Here you can see the various time points. And we can see there is a decline of IgG levels 3 months and 6 months following the second dose in all groups, but particularly in naive participants. But there is an interesting point. The significant difference we observe in IgG levels between the individuals who receive the doses with a long dosing interval and the individuals who receive the doses with a short dosing interval is still maintained even 6 months following the second dose. And finally, I would say as expected, following the third dose, we have a boost of IgG levels in all groups. We also analyzed the antibody responses in individuals who received the AstraZeneca vaccine, so a viral-based vector vaccine. So it's a comparison between the IgG levels of individuals who received the Pfizer vaccine and the AstraZeneca vaccine. Here you have the time points following the second dose, and again it's a comparison between naive and pre-infected individuals. And here you can see that the individuals who received the AstraZeneca vaccine induced lower IgG levels compared to the individuals who received the Pfizer vaccine. But following the third dose, which is a Pfizer vaccine, because in the UK it's always a Pfizer vaccine as a third dose, you can see following the third dose there is a boost of IgG levels in all groups. So now to summarize the main findings of these studies. We found there were higher neutralizing IgG titers in SARS-CoV-2 pre-infected individuals compared to naive participants following a first vaccine dose. We also observed a decline of neutralizing IgG titers during a long dosing interval, particularly in naive participants, but the neutralizing IgG titers are boosted following the second dose. And finally, the main finding of these studies, higher neutralizing IgG titers were measured with a long dosing interval compared to a standard interval, a short interval, in naive participants following a second dose. So to conclude, a pre-infection, the dose regimen, but also the type of vaccine, all these parameters impact on antibody responses post-vaccination. And you can find all results I presented today, but even much more, including T-cell responses, in 2 papers we published in Lancet Microbe and Cell in 2021. And to finish, I would like to thank the PITCH Consortium and all people involved in these studies, and particularly Susanna and Paul who led these studies, and my supervisor, Miles Carroll. And I would like to thank you for your attention.